directly dissolved. And then we can also carry a little bit of carbon dioxide. Carbon dioxide has an entirely different mechanism that I'll talk about on the next slide, but hemoglobin can transport, bound to some of its subunits, a little bit of carbon dioxide here as well. Okay. Now, hemoglobin can also bind protons as well as carbon monoxide. This is where it becomes really, really detrimental. When hemoglobin binds carbon monoxide, it does so at a greater affinity. This is how carbon monoxide is fatal. It actually competes with oxygen for binding of these heme groups on the hemoglobin. And so what we see is that it also binds irreversibly. So when carbon monoxide binds to hemoglobin, it actually doesn't let go. It stays on there. And so you basically suffocate chemically, right? You cannot bind oxygen because all of that hemoglobin is saturated with that carbon monoxide, which binds irreversibly at a much higher affinity than it does for oxygen. Okay, so that's how carbon monoxide can be really, really detrimental in terms of oxygenation. So carbonic anhydrase is an enzyme that facilitates the more common mechanism of transporting carbon dioxide. And so what we see is that carbon dioxide in the presence of water and this enzyme carbonic anhydrase can be converted into carbonic acid. And this is how we can move carbon dioxide around in the blood. We actually move it around in its dissociated form. So it's dissociated here into protons, which are acidic, and bicarb. And then when it gets to the lungs, it is reconverted, right? These double-headed arrows means that this can take place in the forward and reverse direction. So when this bicarb and hydrogen get back to the lungs, they are reconverted to carbon dioxide, which can be exhaled via the lungs. So this is how we shuttle around carbon dioxide in our blood, and this is how it becomes important in that acid-base balance. This is how we can use carbon dioxide to help, or the ventilation of the lungs and carbon dioxide to help with acid-base disturbances, along with the kidneys, which we'll talk about, okay? So CO2 is critical in maintaining the acid-base balance of blood because of this reaction. And you're gonna see this reaction several times throughout this semester, so make sure you remember it. So carbon dioxide in the presence of water and the enzyme carbonic anhydrase becomes carbonic acid, so this is carbonic acid, and then that can freely dissociate into free protons and bicarbonate. Okay. Now looking at the life cycle of an erythrocyte, because they have no DNA, they have no RNA, they have no organelles, they can't make proteins and all that fun stuff, they can't divide via mitosis, they have a very short lifespan. So they are around for only about 120 days. And I'll talk about sickle cell anemia, which significantly cuts down on this time. A sickled red blood cell, or a sickled erythrocyte, has a lifespan of about 20 days, right? And you can see some of the complications that could arise with that significantly shorter lifespan. Now, they're replaced every couple seconds, 2 to 3 million every second, and about 200 billion per day, so really, really fast turnover for erythrocytes. And they're synthesized from the bone marrow via a process called erythropoiesis, right? Erythropoiesis, I'll talk more about, but it's a really important part of generating new red blood cells, and it takes place via a hormone released from the kidneys. Now, after the lifespan of that erythrocyte is up, it is going to make its way to the spleen and sometimes the liver, where it is going to be engulfed and it's going to be recycled, chopped up, broken down. We're going to snatch that hemoglobin off. We're going to do some other things with it and recycle the important parts of that hemoglobin, which is basically the iron. And I'll talk about that when we go forward. So the life cycle is pretty straightforward, right? We'll take that hemoglobin, that erythrocyte, from the bone marrow, we'll circulate it, it'll do its thing, transporting oxygen, and then after 120 days, it'll go to the spleen where it'll die and be recycled. So let's look at the steps in this process. So the first part is erythropoiesis, and this happens under the influence of erythropoietin, which is a hormone. It's a hematopoietic growth factor that is secreted from the kidneys. So it's secreted from the kidneys in response to low oxygen levels. So the kidneys basically are taking a large part of our cardiac output, and the kidneys can detect whether or not there's adequate oxygenation in our blood. And if there's low oxygen levels, the kidneys will then release that erythropoietin, which acts as a hormone, travels into the blood, makes its way to the bone marrow, and stimulates the bone marrow, right? It's a growth factor to make new red blood cells via those undifferentiated cells we talked about. Does anyone remember the name of those undifferentiated cells? The stem cells, yeah. So the stem cells that are in the bone marrow will now be under the influence of this erythropoietin, and they will differentiate down the line of becoming a red blood cell, and that's how we make new red blood cells. So they differentiate to red blood cells or erythrocytes, and then those erythrocytes will go through a process of development where they lose their nuclei, they lose all the organelles. As I mentioned earlier, they do start developing with those things, but by the time they become fully mature, they have lost those organelles and those other components, and they are now producing hemoglobin, that molecule that we talked about, okay? And the hemoglobin is really important in their function of transporting oxygen. Now, they do require a few other things in order to become fully mature or fully developed erythrocytes. They require iron. So iron is a really important component of hemoglobin. We talked about it, specifically the heme part of that hemoglobin. Okay, let's remind ourselves of the structure here. We had those 4 globins, and in the center we had those heme groups, and in the center of that we had that iron. So it's really important in maturing this erythrocyte to its developed stage. Now, we also have different hemoglobin levels for men and women. So men typically around 13 to 18 grams per deciliter, and women about 12 to 16 grams per deciliter. So this is important in our oxygen carrying capacity. There's a lot of theory here as to why there's this difference. It's thought that women have a better capacity. We can carry more oxygen on each hemoglobin, so that's why we have less. There's some other theories about the menstrual cycle, but all we know is that women have less hemoglobin than men do. Some other important components that are needed for the full development: folic acid and vitamin B12. Now, these are required for that developing erythrocyte, so not when it's fully developed, because when it's fully developed, it doesn't have DNA, it doesn't have organelles, but in the process of getting it developed, it does require these components of folic acid and B12. Now, this is not just specific to our erythrocytes. All of our cells need folic acid and B12, but erythrocytes are much more sensitive to this deficiency because they turn over on such a fast rate.